So as Pete has mentioned and introduced me, I'm Sammi. And today I'll be talking to you about our preliminary results from the parr and smolt tagging in River Västerdalsälven, as well as the food webs in the 6 rivers. So our project focuses on understanding the different population dynamics in the Northeast rivers, and basically what is actually driving these changes in populations for Atlantic salmon. And obviously we have different drivers here that we may think will be affecting the population, which includes food availability, habitat characteristics, environmental variables such as temperature, as well as river management, which includes building salmon ladders as well as egg planting. So for this presentation, we'll be focusing on food availability through using our detailed food web data that we've collected, as well as I'll be presenting some of our pit tagging data, which will then help us to understand how the habitat may actually be affecting the population as well. So for our study sites, we have River Västerdalsälven, which is our main study river. So that would be for our pit tagging study, as well as our main study river for the food web study. We've also sampled Hofsau, Mittfrau, and Seilau, and that is also for the food web study as well. So it gives us a comparison between the different rivers. This is a simple food web of what the rivers in the northeast of Iceland would look like. It's very simple, as you can see here. So we have the blue is the juvenile Atlantic salmon, the top, and the black is the insects, and the green is the algae. And these black arrows you see here, these represent energy transfers. So basically, what is being eaten by what, and basically how the energy and resources kind of travel up that food chain. And for this presentation, we'll be focusing on mainly the Kaurmani larvae. As Linner has mentioned, Kaurmani larvae is basically the majority of the food items that we see. And this is the subject that we're talking about here. And basically, this will represent as our main food web linkage to help us understand the food web and give us a sense of the food web, at least for this presentation here. So as you can see here, why we're talking about the Kaurmani larvae is that this is a simple graph on the percentage of food items that are found in juvenile Atlantic salmon in River Västerdalsälven. And we can see that basically the majority of this is from aquatic invertebrates, very little from terrestrial. And more importantly, that most of this is from the Kaurmani family, with 40% of it actually coming from Kaurmani larvae. Therefore, we're going to be focusing on this particular insect for the presentation. So our main research question is, does food availability change over time and space for juvenile Atlantic salmon in the rivers of Northeast Iceland? So the field work and all the sampling has been conducted in the summer of 2020. We collected sites spatially and temporally over from June to August. We collected samples from invertebrates, algae, and chlorophyll, and as well as electrofished, as well as stomach pump, as you can see from the pictures, to collect data on the juvenile Atlantic salmon. And we've also, and then the samples will be processed and analyzed, and then it will be compared over with our long-term data set. And even though we do have these long-term data sets, it is important that we collect these detailed food webs, as that give us a better overview of what the food webs in River Västerdalsälven and in other rivers are actually like. So I'll be going over my results, first off by comparing the different rivers, and then to looking at the data within River Västerdalsälven. I'll also be going through them with like a food web perspective. So I'll be starting with the algae, and then we're going to go up to the Atlantic salmon. So looking at the chlorophyll concentrations in between the different rivers, we can clearly see that Hofsau has the highest chlorophyll concentrations, and that could be supported by the fact that when we were sampling there was a lot more moss and algae, as you can see in the picture there, compared to the other rivers. And as you can see, Mifrau, Selau, and Westerdalsau all had very similar levels. Looking at the invertebrate abundance data, we'll be going through the SERBR data and the DriftNet data. The SERBR data gives us a view of what is on the bottom of the river, whereas the DriftNet data gives us a view of what is on top of a river. So it gives us the full view of exactly what's living in those rivers. So looking at our SERBR results, we can see that River Westerdalsau had the overall highest Chironomidae larvae density. And then as we look at the DriftNet results, we also see that Westerdalsau also has quite a high Chironomidae larvae density, but also that Selau had a very high Chironomidae larvae density. And then when we look at the Atlantic salmon results, this is a comparison of the percentage of stomach items that were found in the different juvenile Atlantic salmon across the different rivers. And most interestingly, you can see that in River Hofsau, they don't only depend on Chironomidae larvae and Chironomidae family, they depend on cumulids, which are black flies, and trichopteras as well. Whereas, as you can see clearly in the other rivers, that Mifrau, Selau, and Westerdalsau all have very high percentages of Chironomidae, and with Selau having the highest percentage of all. So this is the juvenile Atlantic salmon density across the different rivers. And we can see that Hofsau still remains to be very high in the juvenile Atlantic salmon density, and that could also be supported by that high algae density, that chlorophyll concentration that you see before, that there is that really strong food web linkage between the algae and in the juvenile density here. Whereas in Westerdalsau, even though we didn't see a very high algae concentration, and that we saw a very high Chironomidae larvae density, we don't see that much of a higher juvenile density. And that is also due to the fact that it may be that the river itself is highly variable with the different habitats that are found in River Westerdalsau. And it may mean that the food web linkage is not as strong coming from the Chironomidae larvae density to the juveniles. So this is a graph on the number of chironomids that are found in each Atlantic salmon, as well as across their different ages and different rivers. So what you can mainly see is that number of organisms found in each in the salmon depends on the age. So with longer-lived fish, you get more number of chironomids. And you can also see here that Selau also has the highest number of chironomids that are found. And we did see that as well with the DriftNet results, as you remember, that there was also a very high chironomid density. So therefore, this is probably why we're also seeing a very high density found in the stomachs. So this, now we're going to look at the comparisons within River Westerdalsau. So we can see here on the top here, this is a map of the sites that we sampled. And site B here is the highest algae concentration. And then looking at the invertebrate abundance, we can see that site A and site C has the highest Chironomidae larvae density taken using SERBRs. Whereas in DriftNet, we can see that site C itself is the highest for Chironomidae larvae density. So now we're looking at the Atlantic salmon data, and we can see this is the different juvenile Atlantic salmon density across the different sites in the river, and we can see that site B still also has a very high density in juvenile salmon. And we can also refer back to the algae concentration, that there was also a very high algae concentration within site B. But strangely enough, in site C, we noticed that there was a drop in the density when, as you remember, there was a very high Chironomidae larvae density. So it could also show that there's a difference in the food web linkage and how strong these linkages are between sites. So this is also a graph of the number of chiromids that are found across different ages, across different sites. And mainly what you see here is that the salmon diets would shift over time as they grow. And that is mainly supported here, where you can see the number of chiromids found in the stomachs across the time and across different sites. You can see that there are really strong summer peaks, and also that it varies spatially across different sites. So in conclusion, chironomid larvae makes up the majority of the stomach content of juvenile salmon, and that efficiency of energy transfer between the different levels of organisms could be varied between the rivers as well as within the river. And most importantly, food availability is an important driver of population dynamics, but it is also not the only one. And that leads us to our pit tagging data. So pit tagging data gives us a view of what exactly is happening in the river and how the resources are being used over time, as well as giving us an idea of like their individual behavior, and helps us explain some of the variability that we saw beforehand in the results before. So as Pete has mentioned before, these are our pit tagging antennas. So we've installed free pit tagging antennas in October 2019. As you can see here, the blue dots are where the antennas are situated. And in summary, we have tagged 2,980 smolts and juveniles to this date. And for the smolt tagging of 2019 and 2020, we tagged mostly in May and July, and then we managed to tag 1,302 totally tagged over these 2 years. And also, but then we noticed that we only got around a 1% return rate. And you know, it does seem small, but it does coincide with the IC's North Atlantic Group report on Atlantic salmon that is around the rate that we're seeing. And this is why we're using river Vesterdalsau as a model system. So we could get a better idea of what is the density in the river, as well as how much food we may need in the river, and how many smolts we may need in the river to create a sustainable population. And so for this year of smolt tagging, we kind of did things differently. So there was this paper that was published that talks about having a high predation pressure on the last stretch where the smolts migrate out to sea. And therefore, we may think that, oh, maybe something similar may be happening in River Vesterdalsau. Therefore, this year, we tagged around in June and July again, and then we tagged 1,006 individuals. And so this year, we start to release some of them above the lowest antenna. And that actually increased our detection rate from last year. So that is something interesting to look forward to, and something that we will dive further in the future. And looking at our juvenile tagging data, we've totally tagged, in total tagged 638 juveniles, and we've only detected 21% so far. From our detections, we noticed there were in-river migrations, both up and down the river, and some of these detections have also indicated some predation events, and therefore juvenile tagging is starting to give us these evidence that there are predation, and to give us an idea of what exactly is eating the fish, as well as giving us that additional link in the food web to understand a better overview of what the food web is like in those rivers. And we've only really recaptured a few of them over the years, but we've calculated that their average growth per year is around 1.9 centimeters. And therefore, all this kind of leads to the fact that we should do more. We should have more effort in tagging these juveniles, as well as recapturing them, so we can get a better view of the growth and the in-river migration in these rivers. So there's many future work to be had. So as the results you see before in the food web, it's all on the abundance, whereas having the biomass data could give us a better view of the resource use in the river. Therefore, that will be something I'll be looking to do in the future, as well as comparing our data to our long-term data as well. And then, of course, we're linking our food availability data to the habitat mapping data. And of course, doing more pit tagging in the future. And then hopefully our food availability data could be used for the modeling work in which Olivia will be conducting later. So thank you for listening, and thank you for everyone who has helped out with this project as well. Thank you.